Thank you. Thank you for having me tonight. So tonight I'm going to talk about rocky intertidal habitats. If you're here, you probably have already been down to the rocky intertidal, and you know what interesting and diverse habitats they are. There are rocky intertidal habitats that are tide pools and boulder fields and bedrock benches and many different types. And although it's a very rich habitat, one of the things that's sort of defining that habitat is that it's a very stressful habitat. It's unique in the sense that sometimes it's underwater and sometimes it's exposed to air. So species that live there have to be able to tolerate the desiccation that happens when they're in air, the exposure to waves that come in and pound on the shore, different nutrient levels. Plus, because there's so many organisms there, there's a lot of biological factors. There's competition and predation and facilitation. So what this means is that the species that live in the intertidal are uniquely adapted for that particular habitat. One of the things that is characteristic of the intertidal, if you go out and you look at these habitats, is there's very clear zonation. So as this slide shows here, up in the high intertidal, it's really dominated by the physical factors, especially desiccation and temperature up there. And so only a few organisms can really tolerate that. It's the driest area and the most physically stressful. As we move down to the low intertidal, the areas become covered by water more and more often, and so it's less and less physically stressful, until when we get down into the low intertidal here, it's close to being underwater all the time, and we find that biological interactions between the organisms dominate that. So you can see that zonation here. This is a boulder in one of our study sites at Old Stairs on the Malibu coastline. And so because this is a vertical face, you can see the zonation really well. So in the highest parts up here, it's dry. There aren't very many organisms. Up here, you can't really see them, but there's barnacles up in there. Again, it's pretty dry. And then we get to a mussel zone in through here, and then that's in the mid-intertidal. And then as we get to the lower intertidal, the mussels are gone, and we see some algae, and we see some starfish here. I have a lot to say about starfish later today. These starfish only can live where it's mostly wet, so they can't get up very high. Starfish like to eat mussels. And so this biological interaction between the predator and the prey is what's setting this lower limit here, because the starfish will crawl up when the tide is in, and this is underwater, and they'll eat as many mussels as they can, and then when the tide goes out, they'll go back down. And so they can't get up higher, and so that's why the mussels have a refuge up there. This is just a representation of some of the mobile animals that we have in the intertidal. So you've probably all seen shore crabs when you've been out there. We have owl limpets. I'll mention just a little bit about owl limpets before. So some of these, the owl limpets and the chitin down here, even though they move around, they move very, very slowly, and they're typically grazing on the microalgae that lives on the rocks. And then we have starfish down here too. And then there's hundreds of sessile species that never move at all. There's algae and plants. This is I'll mention Endocladia before. Turfweed is the common name, or wireweed. And then this is surf grass down here. And then there are sessile animals. And so this is, again, a little bit different from terrestrial habitats that you might be more familiar with, in the sense that these barnacles up here have cemented their shells to the rock, and they never move. And instead, what happens when they're underwater, the water brings the food to them. So instead of having to walk around or crawl around to try to find their food, the food comes to them. And then we have mussels down here. This is a picture of black abalone on the Channel Islands from the 1980s or so. And you can see that the abalone were so dense there, they're actually stacked on top of one another. So this is supposed to remind me to tell you that we actually don't have very good information about what lived in the intertidal, say, 100 years ago. So people are always asking me, what sort of impacts have there been to the intertidal, and how much have things changed. And there's some anecdotes that people might say, but not really any data until a little tiny bit of data in the 30s and a little bit more in the 40s, and then really not till the 70s when we started really collecting good data. So what that means is it's hard for us to really know how many changes there have been. And it's actually an interesting thing in ecology to try to reconstruct the ecological history of an area and a community. This is actually probably unnaturally high abalone because the abalone were eaten by sea otters, and the sea otters were basically driven extinct from fur hunters around the turn of the century, late 1800s. And so they probably actually are unusually high. So anyway, it's hard for us to understand exactly what's going on, and that's part of the challenge in terms of managing the rocky intertidal habitat, is to try to understand the natural ecological relationships. I suspect everybody here has probably been down to the intertidal, and there's a lot of uses of the intertidal in Southern California. Most people are going for recreation, there's some education, and quite a few people are going down to collect food or bait or something for decoration. So our intertidal is very heavily impacted, and I'll talk a little bit about that later, but the main sorts of impacts that we have is, as I mentioned, collecting for food or for bait, and sometimes poaching in places where people aren't supposed to collect, so there's extraction. And then another thing that people often don't think about very much is that there's a physical disturbance that just comes from walking out in the intertidal, because there's all these sessile animals and plants that when you walk out there, you're walking on them. And if one person walks on them, it probably doesn't cause much impact, but when dozens or hundreds of people walk on them, they're not really all that happy about that. And so there's definitely impacts from that, and from turning over rocks and not turning them back, and from handling organisms. And then finally, we don't have very good information about what the impacts are, but we know that the water quality is not what it used to be 150 years ago. And so there are impacts from pollution too. And as a result, there are a number of species at risk in the intertidal. I just have two examples here. I showed that picture earlier about all the black abalone that were in the Channel Islands, and there used to be a lot of black abalone along the mainland also. And from harvesting and then from withering foot disease, the black abalone are mostly gone. I'll talk about the withering foot disease later. And then we also have owl limpets. This is a pile of owl limpets that result probably from somebody harvesting them for bait, for fishing. So they pop the owl limpets off the rock and they scrape the meat out, and then they use that for bait. And then, as I mentioned, there's impacts from trampling and over-harvesting. So we have this unique habitat, very rich in species, unique species that can only live in that habitat, and we have a lot of use. And so we'd like to protect that habitat and manage it wisely. But we have a big problem in trying to understand how we should manage it, because we really don't understand the dynamics, the interactions of the species, sort of the ecology of rocky intertidal habitats. And as a result, that means that our management strategies often are reactionary rather than informed by knowledge and sort of proactive. And so what I'm going to talk about tonight is sort of a program that we've put in place to try to remedy this. The best example, the most obvious example of a reactionary response to activity in the rocky intertidal is oil spills. So this is a picture of the Exxon Valdez and the oil spill that happened in Alaska in 1989. And almost 11 million gallons of oil was spilled into Prince William Sound and spread around Alaska. And very high wildlife mortality, but also lots and lots of oil in the intertidal. And you can see here that there were attempts to clean up the oil, and one of the things that was done is they used hot water, high pressure hoses, hot water to try to dissolve the oil to get it go back into the ocean where it could be collected, collected in booms and then skimmed up. Turns out that caused a lot of impact, probably way, way more impact to those organisms than would have happened if they just left the oil there. So I actually went up to Prince William Sound a year after the oil spill as part of a team of people who were assigned to go to revisit the whole oiled shoreline and make an assessment about how much recovery there had been since the oil spill. And so I was part of that team. We went to dozens of places on many different islands in Prince William Sound with a team of geologists and Exxon scientists and government scientists. And I was asked to say whether the site had recovered or not. And basically, I had no idea. And that's because there was no before data from the area that was oiled. And so you could go to a place and you could see what was there now, but because you didn't know what was there before, it was really hard to say whether there were impacts or whether there's recovery. We didn't really know much about what was going on. Did not have a good idea about what the impacts of the spill were, and definitely couldn't tell about how well the Rocky Intertidal was recovering from the spill. So that was a lesson to me. I came back to Santa Barbara, where I was at the time, and I started talking to agencies to see if we could do a better job here in California and try to set up monitoring sites that would be permanent monitoring sites spaced along the coast of California. So if we had a big spill like Exxon Valdez, we would be lucky enough to have some of our monitoring sites hit so that we would have before data and we'd have a much better, more scientifically rigorous assessment of what the impacts were from the oil spill and also how well those sites could recover. And what came from that is then the Minerals Management Service, now the Bureau of Ocean Energy Management, or BOEM, and the County of Santa Barbara agreed to fund the beginning of a monitoring network in the County of Santa Barbara. And that grew into this Multi-Agency Rocky Intertidal Network, or MARINE. And so most of what I'm going to talk about for the rest of tonight is data that we've been collecting as part of MARINE. I'll tell you a little bit more about MARINE and how we're collecting the data, and then I'll talk about some of the results that we've found so far. First, I need to acknowledge that MARINE being multi-agency, it is very multi-agency. Whoops, oh, that's funny. Okay, well, on this slide there are like 20 logos instead of 2 logos. I'm not sure why Cal State Fullerton and the US Navy was so lucky as to make it through. If it was UCLA, I would know Julie did something to the slides, but since it's Cal State Fullerton, I just really don't know. Anyway, there are many, many agencies. There are private foundations, state, federal agencies. There's the universities, the Navy, lots and lots of partners. I put up here pacificrockyintertidal.org as a website, so much of the information, actually, yeah, basically most of the information that we collect is available on this website, pacificrockyintertidal.org. And so if you're interested in what's going on in the intertidal and along that coast, you can go to that website. Okay, so what do we do in MARINE? Well, we do actually a lot of things, a lot more than just these 2 things, but we have 2 main activities. The first thing is that we have long-term monitoring that, oh, and I forgot to mention that as part of MARINE, as we grew the network along the mainland, we encompassed other groups, and one of the groups we encompassed was the National Park Service, who about 10 years before had started a monitoring network on the Channel Islands in the Channel Islands National Park. And the Park Service and us were really interested in looking at trends in species abundances over time as an indicator of what was happening to the health of the Rocky Intertidal. So we have one of the main monitoring programs is this long-term monitoring. The other one is biodiversity monitoring, called a bunch of different things, but right now we're calling it coastal biodiversity surveys. And that is a complement to the long-term monitoring, a broader view, but it's so intensive to do that we can't do it very often. Tells us a lot about the community composition and the associations of different species with the habitats. Just in the interest of time, I'm not going to say anything more about the biodiversity surveys, although they're giving us a lot of useful and interesting information too. I'm going to focus on the long-term monitoring. So overall now we've established 236. This number constantly changes, and so that might be a little bit higher now, but 236 sites using marine protocols over the past 32 years. And you can see that they're spread all the way from Alaska down to Mexico. A lot here in Southern California and along the coast of California. That's the highest density, but many sites. The 159 of these are coastal biodiversity surveys. 154 are the long-term monitoring sites, and then we have these other sites in here too. Some of these, 5 sites have been monitored for more than 30 years. 25 sites have been monitored for more than 20 years, 25 additional sites, and 42 sites have been monitored for more than 10 years. So we're really committed to trying to make sure that we can follow these sites over the long term. And I hope when I show you some of the data, you'll see that that's really important, because the Rocky Intertidal, especially because of all those stresses that are influencing species abundances, is a very, very dynamic and complex place. And so it's really important to be able to track these species over the long term. So the main thing that we're doing for the long-term monitoring is we have permanent plots. And we go out either once or twice a year down here. So far we've been going out twice a year to look at the species abundances in these plots. So if any of you have been to any of our study sites, including one right here at Point Fermin, and you've seen these stainless steel bolts that are sticking out of the rock, those are ours. What we decided when we started this is that we needed to have something that was going to last for decades and decades. And so previous people who had set up sites in the Rocky Intertidal used little epoxy markers or maybe little discs that were set in the rock, epoxied into the rock. And when later researchers like myself went back to those sites and tried to relocate where they had done their studies, we couldn't find them. So we put things that you can find and that will stay for a really long time. And they are marking quadrats or irregular plots or transect lines that we use to monitor the different types of species. And for the quadrats, we take a picture of each plot every time we go out. And so we're able to follow the trajectory of these plots through time and collect the data from that. And then this is what plots might look like laid out across the site. This is an aerial picture of one of our sites. This is at Paradise Cove in Malibu. And these are barnacle plots here. These are those endocladia plots, the wireweed. These are mussel plots. These are transect tapes that are laid out to mark off the Seastar plots. And so this is a pretty small site, but that's basically what our site looks like when we're going out to sample it. So what I want to do now is I want to just follow, lead you through 2 plots on one of our sites from the first sample that we collected to almost the last sample that we collected. So on the left here, we have an endocladia plot. So there's a little E. So this is E2, endocladia plot number 2. And on the right here, we have a barnacle plot, B2. So that's barnacle plot number 2. So when we started in 1992, this was mostly endocladia, and this was mostly barnacles. There's a little bit of endocladia in here. There's a little bit of rockweed in here. Sylvicia, you'll see later. So that was in 92. In 93, we can see they mostly look the same in the endocladia, a little bit more rockweed. Barnacles are kind of disappearing here, and the endocladia is coming in. Go to 94. Now this is mostly rockweed. Not really many barnacles. This is really endocladia, even though we're calling it a barnacle plot, but it's really an endocladia plot. It changed on us. By the time we get to 95, all rockweed, can't see anything else, almost all Endokladia. 96, it's kind of the same. 97, sort of the same. 98, the same. 99, the same, except that actually here we're converting over to rockweed. The rockweed is moving through the intertidal and spreading into what were originally barnacle plots and then became Endokladia, and now they're rockweed. By 2000, it's kind of the same, but the rockweed is now beginning to disappear from the barnacle plots. By 2002, the rockweed's completely gone. We got a little bit of Endokladia, still not many barnacles here by 2003. 2004, still all rockweed. We got pretty much the same here in barnacles. 2005, same. 2006, the same, except that all the barnacles have come back in the barnacle plot. Really, just in 1 year, barnacles came back. 2007, rockweed's starting to go away over here. Still got lots of barnacles over there. 2008, not much rockweed left over here. Still got barnacles. 2009, rockweed's completely gone. No rockweed, no Endokladia, not much anything actually. Barnacles over here. 2010, starting to get a little Endokladia back. Still got barnacles up here. 2011, pretty much the same. 2012, pretty much the same. Little Endokladia, lots of barnacles over there. So here's what it looks like, what it looked like in the beginning. This is what it looked like in the end. Barnacles actually looked like they didn't change all that much, but you saw all the changes over that time period that those plots went through. And the Endokladia, if you just looked at these two, you'd think it went from Endokladia to barnacles, but you would have missed completely all that rockweed, all those years when it was 100% rockweed. So that gives you some idea over a long time period how much these plots can change and how dynamic the intertidal area is, even though in a year, if you just went out there, it would just look totally normal. It would just, you wouldn't notice, you wouldn't go out to this site and say, oh, there should be rockweed there. It would just look like the way it should look, but it changes a lot. I'm going to show you some graphs, not a lot of graphs, but a few graphs, and I'm going to start with this one in Paradise Cove to show that that was sort of a long time series. What we're actually doing is from each of those plots, we're estimating what the percent cover, so that's a measure of the abundance, how much cover in the plot was taken up by rockweed or barnacles or rock or whatever. And so this just shows mussel plots, Middleus californianus, barnacle plots at Paradise Cove, and Endokladia plots. And so what you see is in mussels, this is the percent cover, and these are the years. So each one of those graphs or pictures I showed from before would give you one dot here for 1 year. And so the mussels didn't really change very much over this time in Paradise Cove. The barnacles didn't really change very much in this time period of Paradise Cove, but a lot was going on in Endokladia plots. So we started out with quite a few barnacles in those plots. Barnacles crashed, and they never really came back. The Endokladia wasn't really all that common in the beginning. It went up to be very common, and then in 2004, it crashed. In the meantime, mussels, there really weren't any mussels in the beginning. By 2002, 2003, the mussels just took off, and they became very, very common. So looking at one site, what you see is in some of the areas, there's a lot of changes. In other areas, there's just not that much change. Looking right here at Point Fermin, what the data look like at Point Fermin, we established the Point Fermin site in '99. And in the mussel plots, this is mussels up here in the blue. In the mussel plots, we established the plots because there were a lot of mussels there. As soon as, honestly, we didn't do anything. We're not mussel killers. We like mussels, but as soon as we came in there and established those plots, bam, mussels went away. Nothing really came back. Not really that common, and you'll see. I'll show you pictures in just a second. Rockweed plots. Rockweed just kind of was fine up here until around 2005 or so. It started going down, 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 down. So I have a story about rockweed in just a little bit, and this is just one example of what's happening with rockweeds. So this is what the mussel plots, this is an example of a mussel plot that when we first established things at Point Fermin. Look at all those mussels. Where'd they go? They're all, they're just wiped out, right? They're gone. That's actually a bit of a mystery. And then we have surfgrass plots. Surfgrass actually did better after we established the plots, but it varies a lot. And surfgrass, it changes a lot depending on the season. It sort of gets, stressed out by high temperatures or by waves a little bit, and then it comes back and it recovers. So this is pretty normal. We see this in all of our surfgrass plots, a lot of variation, but it's really pretty stable. And then by comparison, our Seastar plots, they started out with a lot of Seastars, and then again, they just crashed and they're staying down low. So, what's the lesson there? Well, the lesson is this is a really complicated system to be studying. This is just a whole series of plots from a lot of different sites, and this, we have hundreds of sites, right? And so this is just a very small section of the sites that we have. These happen to be the ones that we are looking at in LA County. But what you see is that looking across sites, there's really not a very good pattern of, again, this is barnacle cover, so the open circles are rocks and the closed circles are barnacles. So if you look across all the sites, some of the sites just kind of vary around, they drop down and then they come back, and they drop down and they come back. Some go along and then they drop and they don't come back. Some have big drops and then they come back. Some have like these long trends, and then, every single site is different. And that's basically what we're finding all up and down the coast. When I started here, my expectation was that I knew there was a lot of variation. I thought sites that were close to each other would tend to track each other, that the factors that would influence the abundance of, say, barnacles or mussels would be similar in the sites that were close together, and that things might be different in Northern California than Southern California, but things in LA would be more or less the same. Turns out we don't find that at all. Much more complicated than that. There's a lot of changes, a lot of differences from site to site, from species to species, generally not very well coordinated. There are some exceptions. When we have a big event like an El Nino, we can find a signal. So we see consistent changes across many, many sites when we have an El Nino. But just short of something like that, we're not really seeing general patterns. It's just very, very complicated. There are some notable trends that we've seen, and that's actually what I'm going to talk about for the next few minutes. We've seen mostly declines in a number of species, and the ones I'm going to talk about are black abalone, rockweeds, and Seastars. I also want to talk a little bit about what we found out about human trampling, or impacts from trampling and from collecting. And then finally, I just want to say a few words about oil spills before we move on. So let's start with black abalone. I showed you that picture early on of black abalone from the 1980s, and that's when there were a lot of abalone around. Even by the 1980s, there weren't that many black abalone along the mainland, and that's largely because they'd been harvested along the mainland. But right around at that time, in the mid-80s, this new disease that we hadn't seen before came about, was encountered, and it's called withering foot disease. And basically, it's an organism that attaches to the intestines of the black abalone. Whoops, wrong way. To the intestines of the black abalone and keeps it from getting energy from digestion. So the abalone can't feed anymore and basically starves to death. And so as it's starving to death, it gets energy from breaking down its own muscles. And so it's called withering foot because this is a normal black abalone, and this is one that's affected by the disease. And eventually they just get so weak they fall off the rock and they die. This is what an abalone looks like in a crack. So one of the things that I'll talk about in the next couple of slides is that when the abalone go away, the abalone keep the cracks suitable for abalone nice and smooth in there. When the abalone go away, other organisms, anemones and mussels and things go into those cracks. And now abalone can't really get back in there, even if there were abalone around to live there. So this withering foot disease was first discovered in the Channel Islands when the National Park Service was doing their monitoring. And we have data like this for all the different monitoring sites, but this is pretty characteristic. This is for Anacapa Island. And what you see is hundreds of abalone in their plots, including the black numbers are juveniles. And so there's lots of adults, but there's lots of juveniles around too. And then in the mid to late 80s, boom, they just dropped down, and that's when withering foot hit. And basically no abalones out there for many, many, many years. Starting to come back a little bit now at some sites. So what we're able to show from our monitoring along the mainland was when the withering foot syndrome hit the mainland and also how it progressed up the coast. And one thing that's interesting is that even though it's not caused by temperature, it's caused by a bacterium, the temperature does have an influence on it. And when the temperature goes higher, the disease organism is more likely to cause the effects. And so what happened along the mainland is it was first noticed off of Point Conception, a government point here. And you can see we had high abalone numbers the first few samples, and then it dropped off. Then as you go up the coast to Boathouse, you can see the abalone persisted for longer at Boathouse, and then it dropped off. And then the next site upstairs persisted for longer before it dropped off, and then Perissimus persisted for longer before it dropped off. So basically, we watched it as the disease spread along the coastline. As it turns out, it didn't really spread much farther because the temperature starts getting colder, and the organism isn't able to cause the disease so much. But catastrophic declines about 84% or something like that, but in many sites it's more like 99%. The abalone basically are completely gone. We're really getting very, very little recruitment, so very few baby black abalone are coming back into these sites. And even if they did, I showed you that picture of habitat alteration, they likely wouldn't be able to find suitable adult habitat now. Probably the only way to get the abalone back, even if we could get recruits, would be to go in physically and restore that habitat to make it suitable for the abalone again. And so as a result of this, both from the overharvesting and then the disease, the black abalone was listed as an endangered species. And the data that we collected, as well as the data that the Park Service collected, supported that listing, was used in deciding on that listing. The abalone was a pretty obvious case because abalone, they're not really that charismatic, but people like to eat abalone, so people pay attention to abalone and they know when they go away. I next want to talk about decline in a species, rockweed, which really went pretty much undetected until we started examining our data. And part of this is the reason, like I showed you in that first 20-year time series, things like rockweed can come and they go. And so it's really hard to know when rockweed go away whether this is just something that's happening in one place and it's just a natural thing, or whether it's part of a bigger trend. So rockweed are these large fleshy algae, and they occur in the mid-intertidal, like in areas like this. They're really important. They're what we call ecosystem engineers in the sense that they are providing a habitat for many, many other species. I talked about how in the intertidal it's a very stressful environment, and it can be dry and hot. And if you've ever been out and looked at rockweed and lifted it up, what you'll find is that it's wet under there and it's cool under there. So this is the rockweed that normally would be covering this area here, and we've pushed it back to see what's growing underneath. And what you see is there's all these different anemones and chitons and snails and crustose algae and all sorts of things that are living under the rockweed. And so those organisms won't be living there if the rockweed's gone. So what we discovered when we started looking at the data on rockweed was that consistently across all the regions that we were studying along the west coast, that had rockweed, there had been declines. So the amount of the decline varied according to the region. So in the Pacific Northwest, about half of the sites had declines. In the South Coast around here, about a third of the sites had declines. These are just plotting the sites that have declines, so you can see when it happened and how much of a decline it was. In the Central Coast, about 3/4 of the sites had declines. And on the islands, almost all the sites had declines. Now, the declines happen, there's different trajectories on different sites, which is, I think, why we didn't really notice it. If we went out one time and all the sites were missing rockweed, we would have known something was going on. But some sites started missing rockweed back in 1980, and other sites started missing rockweed in 2005. And so there's lots of variation in terms of when it happened, but it's a very consistent loss of rockweed. No clear temporal pattern for when it happened. Variations in terms of where the islands and the central California are the hardest hit. The Pacific Northwest and Southern California not so hardest hit. I didn't talk about the different species, but the different species also, there's a bunch of different species that make up rockweed, and they also have different patterns in terms of which one are the hardest hit and which one are not so hard hit. It's still a mystery why. We're trying to understand why. So we know there's a pattern. We can see that it's happening. We're trying to relate it to temperature or wave activity or nutrients or something like that. I think my best guess is that it does have something to do with changing temperature or changing climates, but it's still a mystery. We haven't really figured it out yet. Okay, so the last depressing decline story that I want to talk about is Seastar wasting disease. So I imagine many of you have read about the wasting disease because it's actually gotten a fair bit of publicity since it's happened, because it's a really uncommon thing to see a marine disease that's so obvious and so widespread and affecting so many different species. So what happens with the wasting disease is that the sea stars will get some lesions, a lesion like this. Then it starts to deteriorate, and then actually it can be in a matter of days, the whole thing just dissolves, and all you're left is a little pile of white goop and some spicules. And nobody knows what the disease is. There have been a bunch of studies, the disease-causing organism. There have been a bunch of studies. There's some papers that are in review right now where they think they've identified the disease. It looks like maybe it's like a two-step thing. Perhaps there's a virus or something that causes the initial infection, and then there's a secondary bacterial infection that actually causes it to fall apart. It's been very widespread, but it's also very patchy. So there will be sites where the sites might be only 5 or 10 miles apart, and one site will have the disease and the other site won't, and then the next site down the coast has the disease again. So it's very patchy, and I'll show you how it's progressed through time, but as time goes on, more and more of the sites that we thought maybe were protected and maybe wouldn't be affected by the disease have been affected. I'm putting this up. You can also get this map from pacificrockyintertidal.org or from seastarwasting.org. And what this does is it's a really nice website if you want to track what's happening with the disease. You can zoom in. I mean, this is a mess right now, but you can zoom in and you can see exactly which sites have been reported to have the disease and which sites have been reported not to have the disease. This is a great example for a great opportunity for citizen scientists too. So there's like protocols and instructions on this website about how to grade the severity of the disease and how to do surveys. And then there's an opportunity to submit your observations. And so you can go out to local rocky intertidals and you can look to see whether you're seeing the disease or not. And so you can do that on the seastarwasting.org. So here's the progression. I don't have the most recent data, but for the first year or so. First seen in sort of spring to summer 2013, and seen mostly around in the Pacific Northwest, mostly around Washington and a little bit in British Columbia. And that's really about it, just a little bit in the Pacific Northwest. By October, we started seeing it all up and down the coast. But you can see, as I was talking about it, being very patchy. It's nothing like the black abalone disease that went to the mainland and then made a very sort of consistent, regular spread across the coastline. We don't know how this spreads. We don't understand what's causing the disease, and so we certainly don't understand how it is spread from one Seastar to the next. But instead of having like a continuous spread as it goes down the coast, it hopped all the way across the whole coastline. And then by February, it really started to fill in a lot. And then and for a while, Oregon was pretty much spared, but then this past summer, Oregon got a lot too. And same thing in parts of Washington that had not been hit very hard previously. They had a warm summer this summer, and it looks like temperature might, again, just like for the black abalone, it doesn't cause the disease, but it might be something that is leading to the proliferation of the disease. We are finding it at about 2/3 of the sites that we're monitoring, something like that. It's very patchy, but there's some sites that are hit very, very hard, and basically all the Seastars are gone from those sites. It looks like maybe there's more prevalence in larger individuals. One of the things that's different about this disease compared to, so there have been other reports of Seastar diseases and other echinoderms in the same group, like sea urchins and some sea cucumbers. What's different this time is much more extensive geographically. Many, many species impacted in the subtidal as well as the intertidal. We never really saw that before. And it continued on in the past when the water temperatures got cool, then the disease sort of dropped out. This time it's continued through cool water, through warm water, through everything. And doesn't show any sign of stopping so far. We're not really sure what the effects are going to be, but we expect really significant ecological effects because I talked about Seastars feeding on the mussels and influencing the mussel zone, but they're really important predators and they have an impact on the whole community. And so when you take all those Seastars off, it's going to be probably really good for mussels and maybe not so good for things that mussels compete with. So we expect really dramatic changes in the community. I don't know how long it's going to take for a recovery. There's some glimmers of hope that Seastars won't go extinct. We're seeing some places where there's a little bit of healing and a little bit of regrowth from Seastars that lost arms but didn't collapse into a pile of goop, and we're able to start to regrow those arms. And we're also seeing some new recruits. So down here in the corner is a graph from one site in Central California. And in blue, it's the abundance of adults, and in red is the abundance of juveniles. And you can see throughout many, many survey seasons, there were very few juveniles found and lots of adults. And then what happened is all the adult Seastars dropped out, but we started to see lots and lots of juvenile Seastars. And so there's some hope. I don't know exactly why that would be. Nobody really understands why there's such good recruitment. But there's some hope that these recruits will grow up, although there are also reports that the recruits are getting the wasting disease too. Don't really know how this is going to play out yet because it's just in the middle of an epidemic. You know, you're observing the epidemic as it happens, and it's too soon to say what the outcome's going to be. Okay, I want to change gears a little bit to talk about human use in the Rocky Intertidal now. I did a study about human use in Santa Monica Bay, and what we did is we studied sites along the Palos Verdes Peninsula and along Malibu, the coast of Malibu. And in each case, what we tried to do is we tried to pair some really high-use sites with low-use sites. And so we wanted to have sites that were similar to each other, except in the type of use that they got. And what we discovered is it's really hard to find a low-use site in Southern California. until I did this study, I didn't really appreciate how just about anywhere you can get has people getting there and looking at the intertidal. I mean, we did find one that doesn't have very high use, one low use place, but here's what we thought was a low use site, and it's got, 15,000 people a year going to visit it. What I show here in red are the sites in Santa Monica Bay, and in blue are sites in other places that have been reported to be really high use. Fitzgerald is a place by San Francisco, very, very popular, and if you're from Central California or Northern California, you think this place is like, the most crammed, packed place you've ever seen, but it doesn't really compare to Leo Carrillo or White Point or Point Fermin here, so we're talking about 55,000 people we estimated. It probably doesn't include the school, it doesn't count all the school kids for sure, so it could be a conservative estimate. So our low use places had 1,600 to 4,300 visitors every year. Made it a little bit hard to tell what the impacts of human use was, because we didn't have a place that didn't have human use. So we can detect differences, I'll talk about in a second, but they're not really capturing all the effects of human activity. Half the visitors just walked through the site, a third of them handled organisms, 6% of them collected organisms, and mostly what they collected were mussels, sea stars, owl limpets, urchins, snails, crabs, and sea slugs. So there's lots of impacts that we detected, and I don't want to show you all the data for that, but I'll just show you that one of the main things that we saw is if it was a large conspicuous invertebrate, it was probably affected by people, mostly by collection. So what we have here on the right are the lower use sites, and on the left are the higher use sites. And it's not perfect because we have some high use sites that had a lot of sea stars, Pysastor crassius. But in general, what we find is that the higher use sites over here had lower abundances, and the lower use sites had higher abundances of things, which is not really all that surprising. It looked like to us, at least from what we could measure, that collecting was actually more detrimental than trampling. So that could help tell you something about how you could manage this habitat. And I'll come back to that at the very end of my talk tonight. But nonetheless, it looked like trampling did have some impact and broad effects on that. And again, our study probably didn't capture the full range of trampling effects because even the low use sites had pretty high visitor use. And then the last thing I want to talk about is oil spills. So I started this whole introduction to why we're doing this by talking about my experiences with the Exxon Valdez. And so a lot of the reason why we set up these sites was so that we would be able to detect effects of oil spills. And darn it, we haven't had an Exxon Valdez size spill yet. And so we haven't really been able to get the full effect of all this work we've been doing. But we actually have had a couple of spills. And so I can tell you a little bit about what we found in that. The first spill that we were able to use our data for was a torch oil spill off of Vandenberg Air Force Base in 1997. And this was not a tanker, it wasn't an oil platform, it was a pipeline that leaked. And it hit some of our sites and it didn't hit some of our sites. And so we were able to use that comparison to try to tease, and it turns out that this happened, there was a 1998 El Nino, and so it happened when there was a lot going on oceanographically and a lot of natural changes. And so one of the things as scientists that we're really trying to be careful about, we want to make sure that we have good evidence if there's an impact, but we also want to make sure that we have good evidence if there's not an impact. So we want it to be, scientifically rigorous. And one of the things that we were able to show is that there really were not measurable impacts from this oil spill. So to the things that we were looking at. There definitely were impacts from the oil, and there are definitely impacts to things like birds, but the Rocky Intertidal, the amount of oil that hit our sites didn't really cause big impacts. So that was interesting. Then we had the Cosco Busan oil spill in 2007 in San Francisco Bay. If you recall, the captain of this tanker, I guess, didn't notice that there was bridge pilings and drove into the bridge piling and had a lot of oil that leaked. And we had relatively recently established sites in San Francisco Bay. And so again, we were able to use our monitoring sites to help determine whether there was injuries from that oil spill. Plus, we had just before this happened, we had established a set of protocols of what you should do to go out to sample a site before oil hits or right after oil hits to try to get a measure of the impact, and also so that you can measure recovery. And so a group of our group from UC Santa Cruz spent a lot of time in San Francisco Bay going around and measuring what was on the sites. We learned a lot. We learned, amongst other things, that the responsible party doesn't always want the scientists on the site to measure the impacts. And so there's a lot of sort of administrative responsibilities that need to be worked out in order to make the assessment. So there's a lot of science issues too, and we're scientists and we like to focus on the science, but in something like this, there's a lot of non-scientific issues that influence your ability to collect the data. But we actually did a really nice job, I think, of collecting information that is feeding into the damage assessment, natural resource damage assessment for this oil spill. Okay, so I want to switch gears here, and that was more a retrospective look at how we've used the data that we've collected so far and kind of what changes we've seen in the intertidal. I want to look forward now and think about what we can expect in the future. And so mostly I expect kind of a continuation of what we've seen in the past, except for the changing climate. So the expectation is that we will have a lot of changes to the marine environment over the next 20 to 50 to 100 years. And these changes are going to happen really quickly and are likely to potentially have some big impacts on the Rocky Intertidal habitats. And so what are these changes likely to be? Well, first of all, and the other thing is that some of these changes are really different from what we expect on terrestrial habitats on land. So we expect increased temperatures, so that's what we expect on land also. Water temperatures and air temperatures. I've mentioned, but I'll mention again, that for Rocky Intertidal organisms, they're exposed both to air and to water. So they're influenced by both changes in temperature, and in many ways, the air temperatures, even though we think of them as being marine organisms, the air temperatures are going to be more important because that's when they're exposed to air, and that's when they're the most stressed. And so as the air temperature goes up, they're likely to be even more stressed. So changes in temperature, changes in sea level. Not really worried about that in most terrestrial habitats, although we are along the coast here very much, but likely to have a big impact on Rocky Intertidal habitats. Decreased pH. So what happens when we increase CO2 in the atmosphere is that much of that gets absorbed by the ocean, not all of it, but a lot of it gets absorbed by the ocean, and that increased CO2 changes the pH of the ocean, is going to make it more acidic. And so I'm not sure how many of you have heard about ocean acidification. Sometimes it's called climate change's evil twin because it's likely to have really big effects, but it really doesn't get very much publicity. So I think not that many people have heard about it. And then possibly increased storm frequency too. And so again, because of the stresses that come from storms, if we're going to increase the storm frequency, it's likely to have, apply more stress to those intertidal organisms. So real quickly, I'm just going to go through a little bit about water and air temperature here. There's a whole range of impacts or changes, rather, that we think are likely to happen from changes in temperature. And so some of the things we've seen already, and some of these are probably going to mostly be seen in the future. I've talked about disease, and I've mentioned how for the black abalone and for the Seastar, it seems to be related to warmer temperatures. And so the expectation, I think, amongst many disease ecologists is that as we get warmer water temperatures in the future, we're likely to get more marine diseases like the Seastar wasting disease. We have started to see some changes in distributions. It's something that in marine we're really focusing much more on, and we're going to try to get better data to track changes in distributions. But here's a case where actually I mentioned that we don't really have much data on what was in these intertidal habitats before there was so much human activity. But here off of Monterey Bay by Hopkins, there was a scientist who collected data in the early 30s, and then Jim Barry and Rafe Sagarin and some others went back 60 years later, and they relocated those transects, and they looked at the same organisms along there to see what changes there had occurred over those 60 years. And basically what they found was that species that were sort of at the more northern species at the southern end of their range went down, and species that were more southern species, and so they were at the northern end of their range, went up. So there were shifts that were consistent with the idea that their distributions are going to be shifting northward. And so it could either lead to an increase or a decrease in species abundances, but definitely changes in the distributions that they thought were due to the temperatures, warmer temperatures. I showed this slide earlier about the declines in rockweeds. I said we don't really know what's going on. I mean, I think the most likely explanation is that it's a temperature effect, but I don't really know. One of my students, Jay Smith, did a really extensive survey of organisms that live in mussel beds. Talked about mussels as being sort of an important, they're kind of like an ecosystem engineer. They provide structure for hundreds of different organisms that live within the mussel beds. If you ever go and you pull apart the mussels a little bit and look in there, you may see some of the things, but there's just a lot of organisms that live there. And so what Jay did is he went and he compared data that he collected in the late 90s to date, and that's in these gray bars here, to data that were collected historically, usually in the 70s, of the organisms in those beds. And so 20 or 25 years difference usually. And he did that all, he did that all up and down the coastline. And pretty much throughout the whole coastline, what he found is that within mussel beds, we just don't have the same biodiversity that we had 20 or 25 years ago. And the overall decline was about 60%. In the Channel Islands, it was 70%. So the Channel Islands, which is a relatively more pristine than the mainland perhaps, but seems to be finding bigger impacts for some of these things that we're looking at. Many, many rare species were gone. Species that were found in the 70s, he couldn't find it at all now. And again, we think it's probably related to ocean productivity and changing climate. Hard to prove that, but that seems like the most likely explanation. Besides the effects of temperature, there's going to be sea level rise, and there's going to be probably increased storms, although the evidence for that is not as clear. It's hard to predict what the sea level is going to change in the future, but the National Research Council recently did a really thorough job for California, and this is their projections, what they think the most likely effect is. And so you can see it doesn't change very much for the first couple of decades and then accelerates. And this is a high estimate and a low estimate. But in general, it looks like 85 centimeters, so about 3 feet of sea level rise by 2100. So that's what the estimate is. For Los Angeles, in 2050, it's about 28 centimeters, in 2100, 93 centimeters. I mentioned we don't really know what is likely to happen on storms, but even if the storms occur at the same frequency and severity that they are right now, they're going to have bigger impacts because the sea level is going to be higher. So bigger impacts on human structures too, for sure, and on other coastal habitats. But for the intertidal, it's likely to mean that those organisms are under more stress, and it's basically shifting the intertidal up higher. So that interacts with this next point, which is what happens to natural shoreline when sea level rise? Well, it retreats, right? It regresses. So basically the coastal cliffs or bluffs, erode, And here's what's happened historically in San Francisco since the last ice age. So this is about 18,000 years ago. This is where the shoreline was at San Francisco. And then as the sea level, after the ice age ended, the glaciers melted and the sea level went up. The shoreline went back and back and back. So it was here about 11,000 years ago, and then 10,500 years ago, and then 8,000 years ago, and then this is the current shoreline. And so we get this regression of the shoreline. That's going to change the beach width, but it's likely to change the area of Rocky Intertidal a lot, in part because if you owned a house out here, you probably didn't want to see the ocean take it away. So what we expect is there's going to be a lot more demand for armoring our coast to protect the bluffs from erosion. And so if that's what's going to happen, in those places, basically the Rocky Intertidal is just going to get squeezed up against the riprap or whatever is used to armor the coast. And the bluffs normally would have eroded away and would have created new Rocky Intertidal. But if we keep that from happening, we're just going to lose Rocky Intertidal habitat. And then finally, ocean acidification. This is something that we have not really considered the full range of effects from ocean acidification for very long. And so there's studies coming out all the time that are indicating new types of effects. Basically, any organism that forms a shell or a skeleton out of calcium carbonates likely to be impacted, especially at the larval stage. They either can't form their shell, or it takes them so much energy to create and maintain the shell that it's going to reduce growth or reproduction. But we're also starting to find things like changes in behavior. So some fish or fish larvae actually in more acidic water have different behaviors that may change their ability to survive. And we expect it to spread throughout the food web because a lot of the organisms that are likely to be most affected by ocean acidification are at the base of the food web. And so if they aren't growing as much, then we may see big impacts on the food webs. And plus, some of the species that are likely to be affected are things like mussels and oysters that are these ecosystem engineers. This is just a list of species that are expected to be vulnerable to ocean acidification. You probably can't read the species, but it just is a wide range of primary producers, from phytoplankton and kelp, and then a whole bunch of different types of animals: sea urchins, mussels, oysters, crabs, corals, coral, and algae. So a big range of species that are likely to change in abundance as the pH gets more and more acidic. So what we expect in the future is we expect that many species will respond to climate change, changes in their seasonal activities or distributions or abundances. We may have new species combinations that don't occur right now because these species are likely to change idiosyncratically. Each species will change a little bit different from every other species. And so we may have new combinations. Unlike the terrestrial ecosystem, where we also expect a lot of changes from climate change, here we expect to have some changes in the sort of the base of the food chain, both because of physical changes in terms of the way the water works and stratification, and also because of ocean acidification. And in general, we actually don't know very much about what's going to happen in the marine environment compared to what we know about in the terrestrial habitat. There's just a fraction of the studies that have been done in the marine environment compared to terrestrial habitats. So there's a lot of unknowns. Okay, so that's kind of a bummer. Sort of depressing, and I was trying to think of a positive spin I could put on that, and I really couldn't do that. We'll just have to wait and see what's really going to happen, but it doesn't really look very good. So I thought I'd end by just thinking about what we might be able to do to try to protect and manage our resources, our intertidal resources. And there's a bunch of things that we can do, but there's some things that are actually relatively simple. And so this is like the EEES, enforcement, education, exclusion, and species restoration. So I'll just go through each of those very quickly. So for enforcement, so when we did our study of visitors in the Rocky Intertidal sites, we spent many hours sitting up on the shore counting people who were coming down, and we watched a lot of illegal activities going on, and we basically saw no enforcement. I think I saw one ranger talk to some people one day at one place. And so that means that although a lot of the activities that are going on are legal, there definitely are illegal activities going on, and we could reduce the impacts to the intertidal if we just had better enforcement. It doesn't necessarily mean law enforcement. I mean, it doesn't mean you have to give people tickets. So this is something that docents could do for sure. And you could focus it just on the areas that had the highest collection rates. It would only help the species that were collected, but it definitely could, it's a relatively simple solution. And I don't know if all of you know about the new network of marine protected areas that's been established along the coast of California. But with that new network of MPAs, there's a lot of activity being focused on those MPAs, and so that's probably an opportunity for better enforcement of collection laws. The other thing that is really important is education. So we know that a lot of education is happening right here at the aquarium. There's even more education of all sorts that could go on. And yeah, we heard how many of the students who come to the beach here have never even been to the beach. And so what they don't know anything about, they're not really going to be likely to want to protect. This is something where we could deal with school groups or organized tours, things like that. And docents, again, could play a really important role, and also signage. So this is probably not that great a sign. Doesn't really tell you very much. It's the type of sign that you see right now, but I know some groups are trying to work on more effective signage that would educate the public about why they shouldn't be taking organisms or what they should be doing to protect them. Another thing that would really help some intertidal areas would be to keep people out of them. So this is kind of a radical suggestion because we really don't do that in the intertidal very much. We're pretty much used to the idea that we're just going to go down and we're going to walk around and do whatever we want in the intertidal, and that's it. But in some places, in other places, people have realized that in order to protect an area, you just need to keep people out. And one of the things that's different, again, about the intertidal is that it's really impacted by just walking on the intertidal. So there are other things, like in areas where there are wetlands, they'll put boardwalks or they'll have signs that say you have to stay on the trail. They don't let you just wander off through the wetland and tramp through the mud and squash all the animals that are living there. And so we may need to think about whether there's places that we would want to restrict access to just pathways, or maybe restrict access to a whole site, so that we would have some areas that were not impacted by people walking out there. That's one of the things that MPAs are trying to do. They're trying to set aside areas so that they can recover more of their natural ecosystem processes. In that case, it's from fishing, and so you can, in the subtidal areas, you don't have to worry about people stepping on the fish or anything. You just have to keep the collection down. But in the intertidal, keeping collecting out isn't enough. So it would reduce impacts from trampling, handling, and collecting. And then finally, there's some opportunities for restoration of target species. I mean, there's opportunities for more wide-scale habitat restoration, but for the most part, it's not the habitat destruction that's causing impacts here, unlike, for example, wetlands that I also work in, where the habitat's been destroyed and we need to restore the habitat. Here, the habitat mostly is there, but the species aren't. And so if we have something like black abalone that has essentially disappeared from our coast, there's an opportunity to try to restore that species. And I think that it would be a challenge for sure. People have spent a lot of time trying to enhance abalone populations, but it's definitely the type of target species that we could restore. So there's a variety of strategies that we can use to try to manage these sites, but you'll see if I look at my EEES approaches here, they all work for harvesting, and some of them work for handling, and the exclusion works for trampling. None of those do anything about pollution. So the pollution problem is something that has to be tackled up in the watershed. And oftentimes we tend to forget that what happens up in the watershed, it seems like it's removed from the ocean, but that water, the pollutants that are in the stormwater or urban runoff are running down into the ocean and having impacts on the coast, on the organisms that live in the coast. So we need to address those sorts of things up in the watershed. And then finally, managing climate change, it's definitely going to be challenging. There's really not as many strategies that we can use to try to avoid the sorts of things like ocean acidification and sea level rise that we might be able to do if we were just talking about trampling and collecting. So people haven't really thought about it very much, and hopefully we can get some brilliant minds focused on this, and we can come up with some innovative strategies. But in the meantime, one thing that I think we're realizing now, conservation biologists, as we think about climate change effects mainly in terrestrial habitats, what we're realizing is the best way to make the communities resilient to the climate change effects is to have ecologically healthy communities. So if we can remove other stressors, we're finding that those species are better able to tolerate, the stress that comes from climate change. And so that's what I think the best thing that we can do to try to protect our communities from climate change is to try to remove all these other impacts so that the species are, doing as well and under as little stress as possible so that they can better handle the changes that are going to come with the changing climate. So I just want to end by acknowledging some of the people who have helped during all this work. Literally hundreds of people have helped with those surveys, many hundreds. There's a bunch of professors and research scientists, many technicians and students who have worked, a lot of agency staff, many volunteers. This is a project that is totally driven by volunteers, especially down here in Southern California. Whether it's students from UCLA or members of the public, we really rely on volunteers for sure. And then I also want to thank the agencies that have funded my work because, of course, I couldn't do it without them. And that's it. Thank you.